Deoxyribonucleic acid, or DNA as it's commonly known, is the fundamental building block of life. This remarkable molecule holds the genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. Think of it as an intricate blueprint, a detailed instruction manual carefully stored within the nucleus of every cell in your body. This blueprint dictates everything from the color of your eyes to your susceptibility to certain diseases. DNA's importance lies in its ability to store and transmit genetic information from one generation to the next. This hereditary information is encoded in the precise sequence of chemical building blocks called nucleotides, which make up the DNA molecule. These nucleotides, adenine A, thymine T, guanine G, and cytosine C, are arranged in a specific order, much like letters in a language, forming the genetic code. The discovery of DNA and its structure revolutionized our understanding of biology and medicine. It opened up unprecedented avenues for research and gave rise to entirely new fields of study, such as genetic engineering and biotechnology. Today, scientists can manipulate DNA to develop new drugs, diagnose and treat diseases, and even modify crops to be more resistant to pests and harsh environmental conditions. The sheer volume of information stored within DNA is staggering. If you were to unravel the DNA in a single human cell and lay it end to end, it would stretch approximately 6 feet. Considering the trillions of cells in the human body, that's enough DNA to reach the sun and back multiple times. This remarkable molecule, with its elegant structure and profound implications for life, continues to fascinate and inspire scientists worldwide. In 1953, James Watson and Francis Crick unveiled one of the most groundbreaking discoveries in the history of science, the double helix structure of DNA. Their model, based on X-ray diffraction data obtained by Rosalind Franklin and Maurice Wilkins, revolutionized our understanding of heredity and paved the way for modern molecular biology. Imagine a twisted ladder, its two vertical rails representing sugar-phosphate backbones running in opposite directions. The rungs of this ladder are formed by pairs of nitrogenous bases, the genetic alphabet of DNA. Adenine A always pairs with thymine T, while guanine G pairs with cytosine C through specific hydrogen bonds. This complementary base pairing ensures that the two strands of the double helix are held together in a stable configuration. The beauty of the Watson-Crick model lies in its simplicity and elegance. The specific base pairing not only explains the structural stability of DNA, but also hints at the mechanism of its replication. The two strands, being complementary, can act as templates for the synthesis of new strands, ensuring that genetic information is faithfully copied and passed on to daughter cells. This fundamental insight into the nature of DNA laid the foundation for our understanding of how genetic information is stored, replicated, and transmitted from one generation to the next. The discovery of the double helix structure was a pivotal moment in the history of biology. It provided a framework for understanding the molecular basis of life and opened up entirely new avenues for research in genetics, medicine, and biotechnology. Watson, Crick, and Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for their groundbreaking work, forever etching their names in the annals of science. Before a cell divides, it must first create a perfect copy of its DNA. This process, known as DNA replication, is fundamental to life and ensures that genetic information is passed down accurately from one generation to the next. But how does this intricate molecular dance unfold? The answer lies in the elegant mechanism of semi-conservative replication. Imagine the DNA double helix as a zipper slowly unzipping, separating the two strands. Each separated strand then serves as a template for the assembly of a new complementary strand. Free nucleotides floating within the cell's nucleus pair up with their corresponding bases on the template strands, following the strict base pairing rules A with T and G with C. This process is orchestrated by a complex molecular machinery, including enzymes like DNA polymerase, which catalyzes the formation of new DNA strands. As the new strands are synthesized, they wind around their respective template strands, forming two identical DNA double helices. Each daughter DNA molecule consists of one original parental strand and one newly synthesized strand. The semi-conservative model of replication ensures that each new DNA molecule receives one complete set of genetic instructions from the parent molecule. This mechanism preserves the integrity of genetic information across generations, minimizing the chances of errors during DNA duplication. The semi-conservative model of DNA replication, while elegant and intuitive, required experimental validation. In 1958, Matthew Meselson and Franklin Stahl devised an ingenious experiment that provided conclusive evidence for this model. Their work, considered a classic in molecular biology, solidified our understanding of DNA replication. Meselson and Stahl grew bacteria in a medium containing a heavy isotope of nitrogen, 15N. As the bacteria multiplied, they incorporated this heavy nitrogen into their DNA. The scientists then transferred the bacteria to a medium containing the normal lighter isotope of nitrogen, 14N. Samples were taken at various time points after the transfer, and the DNA was extracted and centrifuged in a cesium chloride gradient. This technique separates molecules based on their density. DNA containing the heavy 15N isotope would form a band lower down in the gradient compared to DNA containing the lighter 14N isotope. The results of the experiment were striking. After one round of replication in the 14N medium, a single band was observed in the gradient, at a position intermediate between the heavy and light DNA bands. This indicated that the newly synthesized DNA molecules contained a mix of 15N and 14N, consistent with the semi-conservative model. Subsequent rounds of replication showed a progressive increase in the proportion of DNA with the lighter isotope, further supporting the semi-conservative model. The Meselson-Stahl experiment elegantly demonstrated that DNA replication is indeed semi-conservative, with each new DNA molecule inheriting one strand from the parent molecule and one newly synthesized strand.